Welcome to Science Education Channel. In this video we will discussing about milk microbiology. 1. Composition of milk. 2. Sources of microorganisms in milk. 3. Types of spoilage of milk and milk products. 4. Preservation by high temperature pasteurization in detail, UHT, pasteurization test, phosphatase test. 5. Preservation by low temperature. 6. Determining the quality of milk plate count and MBRT. 7. Production of curd, dahi and Shrikhand. 8. Production of cheese, classification of cheese. Production of cheddar cheese and cottage cheese. Milk microbiology introduction. Milk microbiology deals with the study of microorganisms present in the milk and their role or activity in milk and milk products. Milk is an excellent food for human, animals and microorganisms. The contaminated microorganisms grow very rapidly and spoil the milk and make it unsafe for human consumption. Hence, milk microbiology is also concerned with the preservation of milk to prevent its spoilage and to manufacture the various milk products with the help of microorganisms. Definition and importance of milk. Milk is the liquid food naturally secreted by the mammal for the nourishment of their newborn. Due to high nutritive value, milk is an important article of daily diet of vast population on the earth. It gives about 75 to 100 kilocalories of energy per 100 milliliters. Milk is considered as a most nearly complete food for human because it contains most of the nutritional constituents such as proteins, fat, carbohydrate, minerals, vitamins and water. Similarly, it is an excellent nutrient medium for the growth of microorganisms. Composition of milk. Milk is a white opaque complex mixture containing carbohydrate, fat, protein, vitamins and minerals. The carbohydrate, mainly lactose with some soluble proteins and vitamins, forms a water-soluble fraction, miscible solution. Whereas some insoluble proteins, mainly casein and minerals, form water-insoluble fraction, colloidal form, present as an emulsion. In general, milk is composed of about 87.2% water and 12.8% solids. General composition of milk is given below. Iodine and vitamin. Milk is very poor in iron, copper, iodine and vitamin C content. In addition to above major constituents, milk also contains a number of compounds in trace amount. These includes fatty acids, amino acids, amino sugars, sugar phosphates, nitrogen bases, non-protein nitrogenous substances, flavoring substances, sterols, pigments and some antimicrobial substances. Several enzymes present in the milk includes lipase, esterase, phosphatase, proteases, amylase, catalase, ribonuclease, xanthine oxidase, lysozyme etc. The composition of milk varies animal to animals and depends upon the breed of animals, the lactation period, age of animals, the feeding pattern, season and the environment. The comparison between the general average composition of important milking animals such as cow, buffalo and human milk. Sources of microorganisms in milk. When the milk is secreted inside the udder by healthy animal, it is almost sterile. But as soon as it comes out of the udder, get contaminated by microorganisms from various sources. The quantity of contaminated microorganisms depends upon the farm conditions, utensils utilized, handling and processing of milk. The contaminated microorganisms grow very rapidly and spoil the milk within short time. The spoiled milk becomes unsafe for human consumption. The pathogenic microorganisms in the contaminated milk may spread endemic and epidemic diseases. Various sources of microorganisms in milk are as follows. 1. The producing animals. A. Interior of udder. B. Exterior of udder. 2. The milking barn and its environment. 3. Milking utensils and equipment. 4. Milker and handler. 5. Water and other ingredients added into the milk. 1. The producing animal. The animal itself is one of the most significant contributors of contamination of microorganisms in milk. The sources of microorganisms from animals can be from a. Interior of the udder or b. Exterior of the udder. The contamination from interior of the udder reflects health of the animal, whereas exterior of the udder represent teats and animal body. A. Interior of the udder, animal health. The milk recreated by healthy animals inside the udder is usually free from microorganisms. However, when the animal is suffering with some diseases, then the causative organisms enter into the milk depending upon the severity of the disease. The most common disease in milking animals is mastitis, primarily caused by Streptococcus agalactiae, which is normal flora of the udder but non-pathogenic for humans. Other mastitis-causing microorganisms potentially pathogenic to human are S. aureus, E. coli, S. pyogenes etc. The pathogenic microorganisms which may enter into the milk inside the udder include M. tuberculosis, M. bovis, Coxiella burnetii, Brucella abortus etc. B. Exterior to the udder, teats and animal body. Many microorganisms enter into the milk inside the udder through teat opening. Teats usually become contaminated with soil, dung, mud and feeding materials such as wheat, straw, rice straw, grass etc. Similarly, many microorganisms are introduced into the mool from external body of the animals during milking. The most common microorganisms likely to be introduced into the milk from teats and animal body includes Micrococci, Staphylococci, Streptococci, Enterococci, coliforms, Bacilli, Clostridia etc. 2. The milking barn and its environment. Various microorganisms are introduced into the milk from the dirty milking ham, where animals are kept, and its environment. The accumulated excretory materials of the milk animals, mud, feed etc. in the barn, contaminate the milk directly or indirectly. Some microorganisms enter into the milk from dirty environment of the barn during milking. The species of Micrococcal Corumbacteria, Bacillo etc. are likely to enter into the milk from the milking barn and its environment. Therefore, milking barn and its environment should be cleaned during milking. 3. Milking utensils and equipment. The improperly cleaned milking utensils and equipment such as bucket, pail, cans, tanks etc. are major sources of contamination after milking. When the milking utensils are not thoroughly cleaned and dried, there is growth and multiplication of microorganisms on the left out milk residues. These microorganisms are then added into the fresh milk. Improperly cleaned milking machines, device used to milk large number of animals at a time, also contaminate the milk by adding many microorganisms. The most commonly contaminated microorganisms from utensils and equipment includes Streptococcus agalactiae and S. aureus. Other microorganisms include Corynebacteria, Bacilli, Mycobacteria, Micrococci, S. typhi V. cholerae, coliforms etc. Chlorinated hot water should be used to clean the milking utensils, equipment and milking machines to prevent such contamination. 4. Milker and handler. The unhygienic milking by milker and handler introduce many microorganisms in the milk. If the milker and handler is suffering with a disease, may introduce pathogenic microorganisms in the milk. The activities like sneezing, coughing, talking, singing etc. during milking and handling increase the risk of contamination. The bacteria likely to be introduced by milker and handler includes M. tuberculosis, S. typhi, V. cholerae, S. aureus, Diplococci, Shigella species, Proteus species etc. Unhygienic conditions and cleanliness should be maintained by milker and handler during milking to avoid such contamination. 5. Water and other ingredients added to milk. The contaminated water used for cleaning of milking utensils and equipment or add double into the milk introduce large number of microorganisms such as coliforms, fecal streptococci, Clostridia, Pseudomonas, Bacilli, Vibrio, lactic acid bacteria, Corynebacteria, Vibrio cholerae etc. Therefore the water used for cleaning of milking utensils and equipment also used to add in the milk should be properly purified. Certain ingredients added into the milk such as preservatives, sugar, salt etc. also function as the source of contamination. Pathogens transmitted through the contaminated milk. Milk can serve as a carrier for number of human pathogens. The pasteurization process is designed to destroy all types of pathogens in milk. Hence, when the milk is adequately pasteurized, there is no risk of infection of consumer with pathogens. However, if the pasteurization is faulty or post-pasteurization contamination takes place, it becomes risky from public health point of view. The pathogenic microorganism likely to be present in contaminated milk with their source and the type of disease caused are given below in the table. Types of spoilage of milk and milk products. Spoilage of milk. Milk is an excellent medium for the growth and multiplication of microorganisms. Therefore it is spoiled by variety of microorganisms resulting into following. Defects. Food microbiology and milk microbiology. 1. Colored milk. Many pigment-producing microorganisms grow in the milk and produce different specific color shades to the milk which are as follows. A. Blue milk. The growth of Pseudomonas cincinnati produce a blue pigment and develop blue color to the milk called blue milk. This bacteria usually enter into the milk through soil and water. B. Yellow milk. The growth of Pseudomonas cinxanthus produce yellow pigment resulting into the yellow colored milk. This bacteria enter into the milk through soil and water. C. Red milk. The growth of Serratia marchescens results to develop red milk. Development of red color in the milk is associated with the production red pigment, acid and coagulation of casein protein. The bacteria is introduced through water. E. Ropey milk or slimy milk. The growth of certain capsulated or slime-producing bacteria converts milk to a viscous thread or rope-like form known as ropey milk or slimy milk. When the milk is pulled out, there is the formation of gelatinous, slimy thread or rope. The most common bacteria developing ropiness in the milk is Alcaligenes vasculactis. Other includes capsule-producing K. aerogenes K. pneumoniae. These bacteria usually enter into the milk through water. E. Souring and curdling of milk. The growth of some acid-forming bacteria cause fermentation of lactose and accumulation of acid in the milk. Accumulated acids results coagulation of casein known as curdling. This spoiled milk develop unpleasant sour taste and off flavor. The microorganisms responsible for souring and curdling of milk include Streptococcus faecalis, Streptococcus lactis, Lactobacillus species, Micrococcus, Leuconostoc etc. The growth of Cl. perfringens in the milk cause formation of large amount of lactic acid and also gases. This result into the souring, curdling and evolution of gas bubbles in the milk called stormy milk IV, off flavor in milk. Some bacteria, yeasts and molds grow in the milk and brings about certain biochemical changes in the milk resulting into off flavor in milk. For X. A. Streptococci produce excessive lactic acid and results acid flavor. B. The growth of Achromobacter lipolyticum and Candida lipolytica cause hydrolysis of fat and develop rancid flavor. C. The growth of certain molds such as Aspergillus, Rhizophis, Penicillium etc. results moldy flavor. D. Pseudomonas fluorescens produce fishy odor in milk and milk products. Pasteurization of milk. Pasteurization is one of the universally adopted processes for making the milk safe for human consumption and to increase its keeping quality. The name pasteurization is coined after Louis Pasteur in 1884, who applied a heat treatment at 60 to 70 degrees Celsius for wine and beer to destroy undesirable and spoilage organisms. Dr. Soxhlet of Germany in 1886 applied this method for the pasteurization of milk and milk products. Pasteurization is defined as process of heating below 1000 C, 63 degrees Celsius for 30 minutes or 72 degrees Celsius for 15 seconds, to destroy undesirable and pathogenic organisms without affecting nutrient quality of the products. This process is widely applied nowadays for the pasteurization of wine, beer, milk and milk products, fruit juices and other food products whose quality is affected by high temperature. The objectives of pasteurization of milk are: I. To destroy pathogenic and undesirable microorganisms to make the milk safe for human consumption. E. To maintain nutritional quality and to improve keeping quality, preservation time, of milk. Methods of pasteurization of milk. Following three methods are commonly employed for milk pasteurization: I. Low temperature long time, LTLT, or low temperature holding, LTH, method. E. High temperature short time, HTST, method. E. Ultra high temperature, UHT, method. 1. Low temperature long time, LILT, low temperature holding, LTH, method. In this method the milk is heated in a vat, closed metal tank, at 62.8 degrees Celsius, 145 degrees Fahrenheit, for 30 minutes, followed by immediate cooling to less than 10 degrees Celsius. Usually circulating steam of hot water in pipes is used as a medium to heat the milk in jacketed vat vessels. The milk is pasteurized in batches. LTLT method is found to be efficient method of milk pasteurization because it destroys more than 99% pathogenic and undesirable microorganisms without affecting the milk quality. It also increases shelf life of milk. Among the pathogens, M. tuberculosis was considered to be the most heat-resistant pathogen likely to be present in milk, which requires 60 degrees Celsius for destruction. Therefore, previous temperature for LTLT method was kept at 61.7 degrees Celsius for 30 minutes. However, later it was found that Coxiella burnet, Q fever causing rickettsia, may survive this pasteurization temperature. Hence, since 1956 the pasteurization temperature for LILT method was fixed at 62.8 degrees Celsius, 145 degrees Fahrenheit, for 30 minutes. 2. High temp short time, HTST, method. This is a continuous method of pasteurization, in which milk is heated at 71.7 degrees Celsius, 161.1, for 15 seconds and cooled immediately below 5 C. It is also called continuous flow pasteurization or flash pasteurization. The beating of milk is accomplished in a heat exchanger device made up of copper, timed copper or stainless steel. The milk is passed continuously through heat exchanger pipes which are preheated to 71.7 degrees Celsius, and the milk is kept at this temperature for 15 seconds. After heating, milk is cooled immediately below 5 degrees Celsius and then bottled. In recent years this is a most common and widely employed method of milk pasteurization. It also increases shelf life of milk. This method is continuous, rapid and more effective, which saves time, money and most suitable for commercial dairies, large-scale milk pasteurization. The process is economical and saves energy as the principle of regeneration and efficient heat transfer, i.e. heat exchanger, is used in the process. 3. Ultra-high temperature, UHT, method. Milk sterilization. This is a recently developed method of milk pasteurization wherein milk is heated, directly or indirectly, very rapidly in a continuous flow system up to a temperature of 130 degrees Celsius. For 15 seconds or 145 degrees Celsius for 3 seconds, and then cooled immediately to less than 10 degrees Celsius in a vacuum chamber followed by aseptic packaging. The milk pasteurized by UHT method is termed as sterilized milk, because this milk can be stored at room temperature without refrigeration up to 15 days. This sterilized milk has an extremely high shelf life and increases convenience in marketing. This is a very useful method in parts of the world where refrigeration facilities are not always available. There is 100% destruction and reduction of pathogenic and undesirable microorganism in milk by UHT method. Phosphatase test. The phosphatase test is useful to determine whether the milk is properly pasteurized or not. Phosphatase is an enzyme commonly present in the raw milk and is derived from the milking animals. It is destroyed by the pasteurization temperature. Therefore, properly pasteurized milk gives negative phosphatase test, whereas unpasteurized milk gives positive test. The phosphatase test is performed as follows. In a sterile test tube, take 10 ml of milk sample and incubate at 37 degrees Celsius for 5 minutes. Then add 5 ml solution of disodium para-nitrophenyl phosphate, a substrate for phosphatase enzyme, dissolved in carbonate butter, 0.2 m. pH 7.5. Then incubate the tube at 37 degrees Celsius in a water bath for minutes and observe development of yellow color. There is development of yellow color within 15 minutes. Constitute positive phosphatase test indicating presence of phosphate enzyme in the milk sample, and such milk is considered as unpasteurized. Pasteurized milk does not develop yellow color. Principle, mechanism of phosphatase test. Phosphatase enzyme is present in pasteurized or raw milk, which react with disodium para-nitrophenyl phosphate substrate and forms disodium salt of phosphatase, enzyme substrate complex, and liberates p-nitrophenol radical. P-nitrophenol radical develop yellow color to the milk. In pasteurized milk there is no phosphatase enzyme. Hence there is no liberation of p-nitrophenol. Therefore pasteurized milk give negative phosphatase test. The recommended standards for pasteurized milk are as follows. Standard plate count 2, coliform plate count 3. MBRT 4 phosphatase test less than 100 ml more than 4 hours. Negative determination of quality grades of milk. Milk is an excellent nutrient media for the growth and multiplication of microorganisms. Therefore contaminated microorganisms grow very rapidly and bring about the spoilage of milk. The rate of spoilage of milk depends upon the number and types of contaminated microorganisms. Some bacteriological and chemical methods are employed to determine the quality or grades of milk. The commonly employed methods are as follows. 1. Standard plate count, SPC, method 2. Direct microscopic count, DMC, method 3. Dye reduction methods. Methylene blue reduction test, MBRT, 5. Rescissuron test 4. To check proper pasteurization of milk by phosphatase feed. 1. Standard plate count method. This is nutrient agar plate method, commonly employed for quantitative estimation of viable bacteria in milk. The grade or quality of milk is determined on the basis of number of estimated bacteria. To this method, I milliliter of milk sample is serially diluted in the test tubes containing 9 milliliters of sterile distilled water. After dilution, I milliliter of diluted milk sample is platted with sterile nutrient agar medium, cooled to 46 degrees Celsius in a sterile Petri dishes. The Petri dishes are then incubated at 37 degrees Celsius for 24 hours to estimate mesophiles at 10 degrees Celsius for 7 to 8 days to estimate psychrophiles. 3. At 45 degrees Celsius to estimate thermophiles. After incubation, colonies developed are counted and then number of bacteria per milliliter of milk sample tested is calculated by using a standard formula, where N number of colonies counted, D equals dilution factor, V equals volume of milk sample taken. The number of estimated bacteria in milk sample tested is reported as SPC per milliliter. The grades of milk sample determined based on SPC per milliliter are as follows: 1. Very good, excellent. 2. Good SPC per milliliter less than 2 lakhs, SPC per milliliter 2 lakhs to 5 lakhs. 3. Fair SPC per milliliter 5 lakhs to 10 lakhs. 4. Poor SPC per milliliter more than 10 lakhs. Disadvantages: 1. The bacterial count obtained by this method does not represent total number of all viable organisms in milk because certain bacteria do not grow on nutrient agar medium and under experimental conditions. 2. Non-living bacterial count is not known. 3. Some rapidly growing bacterial species inhibit the growth of other species. 2. Direct microscopic count, DMC, method. The total microbial count of milk can be determined by the direct microscopic examination of milk. The method is performed as follows: 1. 1 square centimeter area, 1 cc, is marked on a clean and dry glass slide. 2. 0.01 milliliters of milk sample is spread uniformly over this area with the help of a special pipette or syringe and smear is prepared. 3. The smear is air-dried and stained with methylene blue solution. 4. The slide is examined under oil immersion objective and the number of bacteria per microscopic field is determined. The direct microscopic count, DMC, of the milk sample is calculated as follows. 1. In order to calculate DMC, the area of microscopic field is first determined. Usually the diameter of an oil immersion microscopic field is 0.16 mm, i.e. 0.08 mm radius. 2. Therefore the area of an oil immersion microscopic field, R equals radius. 3. The area over which the milk sample was spread on the glass slide was I square cm minus 100 square mm. Therefore number of microscopic fields in 100 square mm. Area of glass slide is equal to 4. Count the number of bacteria found in one microscopic field. Observe at least 4 to 5 microscopic fields and make the average. For x 8 plus 10 plus 12 plus 10 minus 40 divided by 4 equals 10. Suppose the average microscopic count per microscopic field, y5. Therefore the number of bacteria in 0.01 milliliters of milk is equal to 6. Direct microscopic count per milliliter, DMC, Mi, is then reported as DMC per milliliter. Therefore DMC per milliliter based on DMC per milliliter estimated. The grades of milk determined are as follows. 1. Very good. 2. Good DMC per milliliter less than 3 lakhs, DMC per milliliter 3 to 5 lakhs. 3. Fair DMC per milliliter 5 to 10 lakhs. 4. Poor 5. Very poor, unsatisfactory, DMC per milliliter equals more than 20 lakhs. DMC per milliliter 10 to 15 lakhs. 3. Oxidation reduction tests. When milk leaves the animal body, it is exposed to atmosphere, utensils, handler etc. and get contaminated with various types of microorganisms. The contaminated microorganisms grow very rapidly in milk. Initial oxidation reduction potential of milk is about plus 300 mV. However, when the contaminated microorganisms grow and multiply in milk, there is lowering of oxidation reduction potential. The rate of change or shift in oxidation reduction potential of milk depends upon the number and types of contaminated microorganisms and the rate of their growth. The shift in oxidation reduction potential in milk can be determined by using certain oxidation reduction indicating dyes such as methylene blue and resazurin. These oxidation reduction indicating dyes undergo change in color when oxidized or reduced. The time required to undergo change from oxidized to reduced form is noted, and then the quality and grade of particular milk is determined. Following two oxidation reduction tests are commonly used to determine the quality of milk. 1. Methylene blue reduction test. 2. Resazurin test. Methylene blue reduction test (MBRT) principle. The methylene blue reduction test is based on the principle of oxidation reduction potential. Methylene blue dye, in its oxidized form remains blue-colored but in reduced form become colorless. When methylene blue is added into the milk becomes blue-colored due to oxidized form of methylene blue. But after some time milk turns colorless due to the reduction of methylene blue to a colorless compound called leucomethylene blue. This change in the color of methylene blue from blue, oxidized form, to colorless, reduced form, known as the colorization, and is due to the growth and metabolic activity of contaminated microorganisms. The time required for the reduction methylene blue is correlated with the number and types of contaminated microorganisms in milk. If the number microorganisms in milk are more, it requires less time for the reduction and vice versa. The quality of milk is determined on the basis of time required for the reduction, decolonization, of methylene blue. Procedure. A 10 ml of milk sample is taken in a sterile test tube and 1 ml of diluted methylene blue solution is added into it. The tube is corked or plugged with cotton and incubated in a water bath at 37 degrees Celsius and the time is noted. The tube is observed for the colorization of methylene blue at every 30-minute interval, i.e. 30 minutes, 1 hour, 1.5 hours, 2 hours, 2.5 hours, 3.00 hours, and so on, until the milk become colorless. The time required for the reduction, the colorization of methylene blue is noted. Based on time of the colorization, the quality of milk is determined. Advantage: MBRT is an easy, less expensive, relatively less time-consuming method to determine the quality of milk. Method to determine the quality of milk. Resazurin test. It is another type of oxidation reduction dye test which is used extensively nowadays to determine the quality of milk. Because this test take very less time as compared to MBRT, resazurin, the azoresorcinol, is an oxidation reduction indicator dye which undergoes series of color changes during reduction, i.e. from blue, mauve, pink, colorless etc. based on the shift in the oxidation reduction potential of milk depending upon the number of microorganisms. Procedure. In a sterile test tube, take 10 ml of milk sample and add 1 ml of diluted resazurin solution. The tube is incubated in a water bath at 37 degrees Celsius and observed at regular interval of 15 minutes up to 1 hour. And the change in color is noted. The standard color discs can be used for comparison of change in color. The quality of milk is determined on the basis of particular color change within 1 hour of incubation. Significance: reassurance test considers only the viable organisms in milk. It is relatively simple, easy to perform and very less time-consuming method to determine grade of milk. Shrikhand methods of production, packaging and preservation. Shrikhand is a popular fermented, sweetened, indigenous dairy product having semi-solid consistency with typical sweetish sour taste. It is very popular in the state of Gujarat, Maharashtra and part of Karnataka. It is prepared by mixing chakka with sugar, color, flavor, spices and other ingredients like fruit pulp, nuts etc. to form soft homogeneous mass. PFA. In fruit Shrikhand milk fat on dry matter basis not less than 7.0% and protein on dry matter not less than 9.0%. Traditional method of making Shrikhand. Traditionally Shrikhand is prepared by boiling cow or buffalo or mixed milk and cooled to room temperature, 30 degrees Celsius. Heated and cooled milk is added with previous day dahi at the rate of 0.5 to 1%. Milk is left undisturbed overnight at room temperature to set firmly. It is then stirred and hung in a muslin cloth for 10 to 12 hours to drain off whey. The curd mass obtained after removal of whey is called as chakka. Chakka is then added with calculated amount of sugar, color, flavor and other optional ingredients like fruits, nuts, spices, herbs and served chilled. The chakka obtained from whole milk, standardized milk has smooth body, whereas the one obtained from skim milk is little rough and dry. When whole milk is used for chaks making, high fat loss occurs in whey thereby affecting the recovery of fat in chakka. Therefore, it is preferred to use skim milk for chakka making and then mixing of cream or unsalted butter to adjust the fat in the finished product. Homogenization of milk leads to slow drainage of whey giving high moisture content in chakka and a product with very soft consistency, not liked by the consumers. Conventionally made chakka varies from batch to batch with regard to moisture and acidity. Moisture content affects the yield, consistency and composition, whereas acidity affects the taste and quantity of sugar to be added. Industrial production of Shrikhand. With a view to overcome some of the limitations of the traditional method and to partially mechanize the Shrikhand production, a semi-mechanized large-scale production is employed. Shrikhand is the first traditional milk product for which large-scale production technology was adopted. The first modern plant has been established at the Baroda District Cooperative Milk Producers Union Limited. Baroda Dairy has adopted a process which involves use of basket centrifuge for speedy draining of whey and a planetary mixer for kneading and mixing of ingredients for industrial production of Shrikhand. Fresh skim milk is used as a raw material. Use of skim milk has got many advantages as listed below. Fat losses are eliminated. Faster moisture expulsion, less moisture retention. Skim milk is heated to 85 degrees Celsius for 30 minutes. Cooled to 30 degrees Celsius and inoculated with LF40 culture containing Lactococcus lactis subsp. Lactis and Lactococcus lactis var diacetylactis at the rate of 1.0 to 1.5%. After the required acidity of 0.8 to 1.0 is reached, the curd is taken into basket centrifuge or quark separator to remove whey from the curd. The curd mass or chakka is taken into planetary mixer or scraped surface heat exchanger. Sugar at the rate of 80% with W. Calculated amount of plastic cream, 80% fat, to give at least 8.5% FDM in the finished product are added and mixed thoroughly. Optional ingredients like color, flavor, fruits, nuts etc. can also be added at this stage. Then it is packed at room Temperature and stored at refrigeration temperature. Packaging of Shrikhand is a semi-solid product. Heat-sealable polystyrene cups of 100 grams to 1000 grams capacity are commonly used for packing Shrikhand. However, small manufacturers sell the product in wax-coated paperboard boxes. Shelf life of Shrikhand. Due to both high acid and sugar levels, Shrikhand has a fairly long shelf life of 30 to 40 days at 8 degrees Celsius and 2 to 3 days at 30 degrees Celsius. The shelf life depends largely on the initial level of contaminating organisms, particularly yeast and molds. The shelf life of Shrikhand can be increased by addition of potassium sorbate 0.05% or by thermization at 65 degrees Celsius for 10 minutes coupled with addition of 0.02% sorbic acid. For industrial purposes, pasteurization of Shrikhand at 65 degrees Celsius, 10 minutes and subsequent freezing can increase the shelf life up to 12 months at -26 degrees Celsius. Introduction. Consumption. Fermentation gives an acid taste to milk which is particularly refreshing in WCEM climate and also imparts certain therapeutic benefits originally absent in milk. Fermented dairy products have assumed prominent position in human diet in many regions of the world. Fermentation leads to partial breakdown of milk constituents and increases the digestibility of cultured milk products. Dahi preparation methods, quality of dahi, packaging. Shelf life and defects. Fermentation is one of the simplest ways of preserving milk constituents for humans. Some of the popular Indian fermented milk products are dahi, lassi, chakka, Shrikhand, mishti dahi and raita. Product description. Dahi is produced from heat-treated milks after inoculation with certain species of lactic acid bacteria added to milk in the form of starter culture. Lactic acid bacteria added mahipli, grow and produce lactic acid, acetic acid and carbon dioxide by utilizing lactose present in milk. Some bacteria uses citric acid of milk to produce certain volatile organic compounds, mainly diacetyl, which is mainly responsible for flavor of dahi. Judicious combination of acid-producing and flavor-producing microorganisms in the starter helps in the production of dahi with a firm body and good flavor. Definition of dahi. Dahi or curd is a semi-solid product, obtained from pasteurized or boiled milk by neg, using harmless lactic acid or other bacterial cultures. Dahi may contain additional cane sugar. It should have the same minimum percentage of fat and solids not fat as the milk from which it is prepared. Where dahi or curd, other than skimmed milk dahi, is sold or offered for sale without any indication of the class of milk, the standards prescribed for dahi prepared from buffalo milk shall apply. Method of preparation. Traditional method. In traditional method of dahi preparation, milk is heated intensively to boil for 5 to 10 minutes and then it is cooled to room temperature. Cooled milk is added with previous day curd or buttermilk, stirred and allowed to set undisturbed, usually for overnight. At Halwar's shop, milk is considerably concentrated before being inoculated with starter culture, so that the total solid content of milk gets increased, particularly increase in the protein content of milk. Concentration of milk results in custard-like consistency of dahi and keeps the product from weighing off. Industrial method of making dahi. Selection of raw material. Production of cultured, fermented milk demands high-quality raw materials with respect to physical, chemical and microbial standards. Filtration, clarification. Fresh raw milk is heated to 35 to 40 degrees Celsius to aid clarification or filtration process, then it is filtered to ensure that milk is free from extraneous matter. Standardization. Fat, 0 to 5%, SNF, 11 to 13% fat is standardized based on type of product ranging from fat-free to full fat and SNF. level is increased by min. 2% than that of milk. It is common to boost the SNF content of the milk to about 12% with the addition of skim milk powder or condensed skim milk. Increased SNF in turn increases the protein, calcium and other nutrients and resulted with improved body and texture, custard-like consistency. Higher milk solids prevent weighing off of the product during storage. Homogenization, 175 kg per square centimeter. The standardized milk is subjected to homogenization after heating to 60 degrees Celsius to increase the efficiency. Homogenization reduces the cream layer formation during incubation. Single-stage homogenization with 175 kg per square centimeter pressure would be sufficient to improve texture of the heat treatment. 9. C. 23 minutes. Milk intended for dahi or any other fermented milk product is given severe heat treatment L. 90 degrees Celsius for 10 minutes following. Other benefits of high heat treatment. 1. Denatures and coagulates milk albumin and globulins which enhance the viscosity and produce custard-like consistency. 2. Kills contaminating and competitive microbes. 3. Development of relatively sterile medium. 4. Removal of air from the medium to more conducive for the growth of culture bacteria. 5. Effective thermal breakdown of protein-releasing peptones and sulfhydryl groups. This in turn provide nutrients to starter bacteria. Packaging and fermentation. The heat-treated product mix is cooled to 37 degrees Celsius and it is inoculated with specific dahi culture at the rate of 1 to 1.5%. Starter culture is the most crucial component in the production of high-quality fermented milks. Proper selection of culture strains decides the good quality of product. Dairy cultures are available in various forms like freeze-dried, liquid and frozen forms. After the product mix is inoculated with dahi culture, it is thoroughly mixed and filled into plastic cups. Scaled properly to avoid any contamination and spillage of the product. Dahi is packed in food-grade polystyrene and polypropylene cups in 100 grams, 200 grams and 400 grams pack sizes. Various packaging machines of up to 400 cups per minute speed are available to package cultural dairy products in different sizes. The packaged product should be stored at less than 5 degrees Celsius for extended shelf life. Thus packed product is arranged in cases or crates and transferred to incubation 100 meters maintained at 37 degrees to 42 degrees Celsius. The product mix is incubated till its pH reaches 4.4 to 4.5 and then it is cooled rapidly to less than 5 degrees Celsius by exposing the cups to high-velocity cold air storage. Dahi is normally stored at 4 to 5 degrees Celsius. Storage area should be maintained clean and tidy to avoid any cross-contamination. Cheese introduction. Cheese is one of the oldest milk products known to mankind. Ancient people have learned to prepare cheese as a means to preserve milk. Now at least 400 varieties of cheese are known in different parts of the world. However, there are common 20 types. About 40% of the milk produced in the world is utilized for cheese manufacture. In 2005, the world cheese production was noted to be 20 million tons. Definition: cheese is a concentrated, moisture-removed, milk product made from curd obtained by coagulating the casein with the help of rennet enzyme or lactic acid-producing bacteria and then salted and ripened. By holding it at suitable temperature and humidity. Types of cheese. Cheese is divided into two main groups, based on the process of curdling the milk involved in cheese preparation. I. Acid curd cheese or cottage cheese. I.e. rennet curd cheese. I. Acid curd cheese. This type of cheese is prepared by curdling the milk by inoculating lactic acid-producing bacteria, e.g. Lactobacillus bulgaricus, or directly the lactic acid. Rennet curd cheese. This type of cheese is prepared by curdling the milk by the action of rennet, renin, enzyme. Further, on the basis of moisture content, physical state, cheese is classified into 3 types. 1. Soft cheese, having 55 to 80% moisture. 2. Hard cheese, having 30 to 40% moisture. 3. Semi-hard cheese having 40 to 55% moisture. The cheese is refined by using bacterial species or mold species. Therefore, cheese is also classified into 2 groups based on the type of microorganisms used for refining by I. Bacteria-ripened cheese. I. Mold-reopened cheese. Given below in a table. The summary of classification of cheese based on different production of cheddar cheese. The manufacture of cheddar cheese includes the process of cheddaring, which makes this cheese unique. Cheddar cheese is named for the village of Cheddar in Somerset, in southwest England, where it was originally manufactured. The manufacturing of this cheese has since spread around the world, and thus the name has become generically known. Food ingredients used during manufacture. Milk, in general. The milk is raw milk, whole or 3.3%. The milk must be ripened before adding in the rennet. The term ripening means allowing the lactic acid bacteria, lab, to turn lactose into lactic acid, which lowers the pH of the solution, greatly aiding in the coagulation of the milk. This is vital for the production of cheese curds that are later formed into cheddar. Rennet, chymosin, renin. Rennet is an enzyme that was traditionally collected from the stomach of a milk-fed calf, natural rennet. This enzyme is responsible for the coagulation of the milk proteins to produce curds. Cheese produced this way is neither vegetarian nor kosher. Coagulation can also be achieved using acids, but this method yields lower quality cheddar. The 2 key components of natural rennet are chymosin and bovine pepsin. Extracts from plants such as nettles were found to produce similar effects and have been used in some types of cheese making. Vegetable rennet. When calf rennet grew scarce in the 1960s, scientists developed a synthesized type of chymosin by fermenting certain bacteria or fungi, microbial rennet, but this also was not useful for all types of cheese making. A solution using recombinant gene, GMO microbial rennet, technology was developed and approved by the U.S. Food and Drug Administration in 1990. This splices the calf gene for producing chymosin into the genes of certain bacteria, yeasts or fungi, producing pure chymosin. Equipments required. Stainless steel knives are used to uniformly cut the curds at various points during the process. The device is a stainless steel frame with stainless steel wires, which cuts the curd cleanly. Vats will vary depending on a variety of factors, but in general are in the shape of an oval or rectangle with hollow walls that are used for holding warm water to keep the curds at desired temperatures. This part of the vat is referred to as the jacket. A milling machine is used to cut the matted curds that are formed during the cheddaring process. This allows the curds to be easily salted. Process. Add rennet. In general, 85 to 115 g 3 to 4 ounces of rennet is added per 450 kilograms, 1000 pounds, of mix. The wreath will serve to coagulate the milk protein and form curds. The vat must be mixed thoroughly after the addition of the rennet to ensure equal mixing. And it also helps to dilute the rennet to make it easier to spread around. Pure water is used when diluting the rennet, as any pH that is not near 7 will rapidly decrease the effectiveness of the rennet, as will chlorine. Thus, water with impurities will result in a vastly decreased yield of cheese. Setting the curd. Once the rennet is added, the mixture must be allowed to set and form curds. The mixture is kept at around 29 to 31 degrees Celsius, 84 to 88 degrees Fahrenheit. Temperature is controlled by flowing warm water through the jacket of the vat. Setting time varies, and allowing a proper amount of time is vital. It takes anywhere from 30 to 40 minutes to set the curd. The most common way to determine when the curd is set is by inserting a flat blade at a 45-degree angle into the curd and raising it slowly. If the curd breaks cleanly leaving a glassy fracture, it is ready for cutting. In a large cheddar manufacturing facility, this may also be tested using a viscometer. Cutting the curd is cut into 60 or 16 mm D in cubes using stainless steel wire knives. A smaller cube size means the cheese will be lower in moisture, whereas a larger cube size will result in a high moisture cheese. It is important that cutting time is minimized and that the cuts be clean. The best way to determine how efficient the cutting job was is to determine the fat content of the whey. The optimal level of fat content of whey is 0.3% fat or less. The curds are handled gently after cutting to prevent fat and protein loss to the whey. The curd is prevented from sticking to the sides of the vat, but minimal agitation is desired. The curds are allowed to set again for 10 to 15 minutes. Fat and protein loss may affect the ability of the cheese to be considered cheddar cheese, depending on the region where it is created. Cooking the curd is cooked by adding hot water to the jacket of the vat, up to 39 degrees Celsius or 102 degrees Fahrenheit. The curd is stirred constantly during this step to avoid uneven cooking or overcooking, and the cooking will only take 20 to 60 minutes. The whey's pH will be around 6.1 to 6.4 by the end of the cooking. Draining the curd. Initial piling of the curd. In this instance the curds were piled and allowed to sit before being cut, and separated whey is removed from the curds by allowing it to drain out of the vat. In general, a gate is present to prevent curds from escaping. When most of the whey is gone, the curds are raked to either side of the vat, allowing whey to drain down the middle of the two piles. Cheddaring. Cheddaring is a unique process in making cheddar cheese that involves stacking loaves of curd on top of one another in order to squeeze additional whey out of the loaves below. It is a multi-step process that reduces whey content, adjusts acidity, adds characteristic flavor, and results in a denser and sometimes crumbly texture. Setting. The curds are allowed to set until they reach a pH of about 6.4. Cutting. Loaves of curds are cut about 15 centimeters, 6 inches, wide along each side of the vat. After 10 minutes, the loaves are turned over and stacking begins. Stacking loaves. Every 10 minutes when the loaves must be turned over, they are stacked. This step uses the weight of the loaves upon each other to help expel additional moisture. The first time this occurs, 2 loaves are stacked together. The next time, the loaves are turned, and 2 stacks of 2 are put together. When the stacks get large enough, in general, 4 high, stacking stops, but the loaves are still turned every 10 minutes. This process is complete when the acidity of the whey is between 5.1 and 5.3, so it is checked constantly. Milling the curd. When the turning process is complete, the loaves must be cut down into a size that fits in the mill. The mill will cut the matted curd into about 1.3 centimeters D in pieces. During this process, the milled curds are constantly stirred to avoid rematting. Salting. When all of the curd is milled, salt must be added. The amount of salt varies, but it will be between 1% and 3% by weight. The salt must be mixed thoroughly. Salt helps remove some of the whey from the cheese, which lowers moisture content, adds to the flavor of the cheese. and will also stop the cheese from becoming too acidic, which imparts a bitter taste. Packaging and pressing. The curds are placed into molds that will be used to press the curds and form the blocks of cheddar. After this, the cheddar cheese will be aged. Aging. Aging time depends on the type of cheddar. Aging. Aging time depends on the type of cheddar. In general, mild cheddar is aged for 2 to 3 months. While mature, sharp, and extra mature cheddar is aged 12 to 18 months. The oldest cheddar has generally been aged about 10 years. Cottage cheese introduction. Most of the cheeses are made from either whole or skimmed milk. Paneer is an ancient indigenous product in India. The milk from cow, goat, sheep, mare, camel and other domestic animals is used for cheese preparation. Cottage cheese is an unripened acid curd soft cheese with mild acidic flavor, commonly prepared in America as a household process. Cottage cheese is thought to be the first cheese made in America. Preparation of cottage cheese. Cottage cheese is an acid curd, unripened soft cheese having moisture content, about 70%. It is usually prepared from cow's skim milk. The various stages involved in the preparation of cottage cheese are as follows: 1. Curdling the milk. 2. Processing of the curd to remove moisture. 3. Salting. 4. Aging, ripening. 1. Curdling of the milk to prepare cottage cheese. A good quality pasteurized cow's milk is used. It is then inoculated with 5% starter culture of lactic acid producing bacteria such as Lactobacillus lactis, Lactobacillus bulgaricus, Streptococcus lactis etc. The milk is then heated gently at 35 to 37 degrees Celsius for about 6 hours. During heating, inoculated starter culture of bacteria grow and multiply vigorously in the milk and produce more and more lactic acid. The initial pH of the milk, which is usually 6.8, gradually decreases and reaches up to pH 4.6 to 4.4 due to accumulation of lactic acid produced by starter culture. When pH of the milk drops to about 4.4, the casein protein in milk gets coagulated and precipitated. Coagulation and separation of casein protein from milk is known as curdling of milk. The watery fluid which separates out during the formation of curd is called as whey. It consists of 93% water, 5% lactose and small amount of minerals, vitamins, albumin and fats. Actually, casein remains as Ca caseinate in fresh milk during curdling. Lactic acid produced by inoculated organisms reacts with calcium ion and forms Ca lactate. When the casein content gradually decreases due to the formation of more and more Ca lactate with accumulated lactic acid produced by lactobacillus and reaches to a certain minimum level, at the pH range 4.6 to 4.4, the casein gets separated, coagulated and precipitate out. The straw-colored watery fluid known as whey separates out from the coagulated casein. The whey contains about 93% water, 5% lactose and other water-soluble vitamins, proteins and minerals. Whey is a nutritious byproduct of cheese preparation. 2. Processing of the curd to remove moisture. The casein separated from the whey during curdling is processed further to remove moisture content. The moisture, water, is removed with or without applying the pressure. The amount of moisture to be removed depends upon the type of cheese to be prepared: soft, hard or semi-hard. To prepare soft type of cheeses such as cottage cheese, the moisture content is maintained in between 65 to 80%. Therefore, only 20 to 35% moisture is removed from the curd. To prepare cottage cheese, the moisture is removed without applying the pressure. For this, the curdled milk is placed in perforated trays to drain out the water. When the water content is removed to a desired level, the curd attains firmness and forms a flat cake-like texture. The formed cake is then cut into the pieces of desired shape and size depending upon the type of cheese to be prepared by a special curd knives or wire knives. The pieces are then cooked, heated, slowly at about 40 to 45 degrees Celsius for 30 minutes. Cooking helps in further expulsion of moisture from curd pieces, cubes, and to develop more firmness and texture in cheese. 3. Salting sodium chloride. Sodium chloride, as a salt, is added to the pressed, moisture removed, and sized curd cubes, known as salting. Salting may be done by adding dry salt over the surface of the pressed and sized curd cubes. In some varieties, the cheese block is dipped in salt solution, brine, or dry salt is rubbed over the surface. About 2 to 3% of salt is added for salting the cheese. Advantages of salting. A. Salting suppresses, inhibit the growth of undesirable microorganisms, and also check the growth of starter culture bacteria. B. Salting also brings about the certain physico-chemical changes in the curd and in the development of taste and flavor. After salting, the curd is filled in heaps and pressed to remove excess weight to form a definite shape cheese blocks. 4. Ripening of cheese. Ripening, also called as curing, maturing or aging, it is a process of cheese preparation carried out under controlled conditions of temperature and humidity to bring about desirable changes such as taste, texture, flavor, etc. In cheese, the period of ripening varies from 3 to 24 months depending upon the variety of cheese. The ripening changes are brought about by starter culture and other organisms inoculated for ripening. Certain physico-chemical and biochemical changes occur during ripening of the cheese. The body of the cheese, curd, changes from tough and rubbery to mellow and elastic, while the texture becomes more compact during ripening. The fresh cheese has mild acid taste and aroma, which changes to typical flavor in ripened cheese as a result of production of several flavor compounds through biochemical and enzymatic action of microorganisms. However, the cottage cheese is a type of unripened soft cheese in which ripening process is not involved. It is eaten fresh without ripening. After salting of the cottage cheese, some preservative and cream is added so that its fat content is raised to about 5 to 10%. This improves taste, flavor and texture of cottage cheese. It is then packed and marketed. About 10 to 15 pounds of cottage cheese is obtained per 100 pounds of cow's skim milk. Cottage cheese possesses rather poor keeping quality because of its high moisture content, which favors growth and multiplication of contaminated microorganisms. It gets spoiled easily and produce disagreeable flavor and taste. Therefore, cottage cheese is eaten fresh within 2 to 3 days of manufacture. Cottage cheese is high protein product and is a good source of riboflavin and calcium. Some cottage cheese products are fortified with calcium. Storage and handling. Cottage cheese is stored in a closed container and kept in the refrigerator, typically set at 4 to 6 degrees Celsius. Cottage cheese is stamped with a sell-by date, which refers to how long the retail store can keep the product for sale. If any mold growth occurs on the surface of the cottage cheese,